So our last macromolecule that we'll discuss are nucleic acids. We'll go ahead and zoom in. So the nucleic acids serve as information systems in the cell. The DNA is a storage system, RNA is a delivery system, and RNA has a host of other properties or roles in the cell that we'll get into as we move along. So DNA and RNA, they definitely continue with our monomer-polymer motif, and I'll talk about that here. So here would be a polymer or a polynucleotide, and it's composed of these units. The nucleotide comes in several kinds, and we'll talk about the different nitrogenous bases. You've probably heard about A's, G's, C's, T's, and hopefully U, and we'll define those in a minute. The nitrogenous base is sort of like the R group of the amino acids. They're hooked up to a sugar. The sugar is different depending on whether it's DNA or RNA. For RNA, it's ribose, and for DNA, it's deoxyribose, and that oxygen is missing from this position here. I'll show you that in a closer picture in a minute. That's hooked to a phosphate group. When you're making your DNA, which we'll talk about in a later chapter, this actually comes in as a molecule with 3 phosphates, and the cleavage here is the energy to drive the reaction to make it happen. I mention it now because we're going to talk about it again later, so might as well bring it up here. There's a direction to DNA, and it's a direction based on the numbering of our carbons. Remember we talked about carbohydrates and sugars? There was the carbons, and any organic molecule for that matter, there's an order. You number the first carbon, second, third, fourth, and here's our 5th. You're going to hear 3' to 5' often. The prime refers to the fact that it has a modification on it. So if we look at the bottom, the 3' end, it has an OH hanging off it, because remember we are going shorthand, we're not putting all those in here. Then there's the 4th carbon, and there's our 5th carbon, and that's the one that gets attached to a phosphate, and then that phosphate attaches to this OH on the next one. So here's our different units. We're going to repeat this idea 3' to 5' again later on when we talk about copying the DNA and how picky the enzymes are for directionality, which is actually very important because it makes the molecule in the correct way. But just I'm going to point it out here so you know. They call this the sugar phosphate backbone because it's a repeating units of sugar, phosphate, sugar, phosphate, sugar, phosphate, and then the bases hang off in the center. So what are those bases? I already told you about the differences in the sugars. DNA has adenine, thymine, guanine, and cytosine. And in RNA, one of the other differences between RNA and DNA is that instead of having a T, you have a uracil, a U. There's other things between them. Typically you find DNA is a double-stranded molecule, RNA is a single-stranded molecule that can fold up on itself, but there's even more stuff, so we'll deal with that again later. So let's get a little bit closer to the sugars first. I mentioned before ribose has the oxygen in that second carbon attached to the second carbon. Deoxy, without an oxygen, is missing. The only difference between them. They make a pentagon because the oxygen gets incorporated into it and then it throws that 5th carbon up there. Alright, so now let's look at the bases. Unfortunately, they do have specific names for them. Is it important to remember it? I kind of like to, because then when you're discussing things you actually have the vocabulary to know what you're talking about. As with a lot of stuff in biology, you're going to find you're going to be learning a whole nother language almost, and it even gets worse when you get into different disciplines like immunology, but we won't have to worry about that until later. So the pyrimidines, I remember them because they have a Y in them, and cytosine and thymine, the ones in DNA, both have Y's in them too, so pyrimidines and thymine, cytosine. And then since uracil replaces the T in RNA, I know that uracil is also a pyrimidine. They have a single ring, heavy and double single bonds, lots of action going on here, and they're nitrogenous bases because they have quite a few nitrogens in them. I also remember pyrimidine is the longer name, and these are the single ring structures. The purines, on the other hand, are A and G. They are a double ring structure, and they have the shorter name. Don't know if that works for you, but it works for me. So, how do we find out about DNA? Well, we'll go through long sets of experiments in a month or two, maybe two, about how DNA was proven to be the molecule that is containing the heritable information. But suffice it to say, by the late 40s, getting close to 1950, biochemists knew that DNA was very important. And there was a race, literally, to figure out the structure first. It was very competitive. Scientists work together, but they also very competitive. And there's lots of that went on in this story. Watson and Crick are the two scientists that you generally hear about the most. Watson is an American who went to study in Cambridge, and he met Crick there. They're very theoretical chemists. They like to build models, like that's in this famous picture here, and try to figure out how DNA was built based on building structures and theorizing how things went together. Maurice Wilkins and Rosalind Franklin, on the other hand, were the two who did the work to prove that it was a helix. I zoom in on Rosalind because she did not get the Nobel Prize with the other three, because by the time they awarded the Nobel Prize in 1962, she had passed away from ovarian cancer. Unfortunately, she probably got the cancer because of the exposure she had to X-rays during her career as a chemist. Solving structures. She solved the structures of viruses, which we'll talk about again later on in the year, and she also helped to solve the structure of DNA. To do that, you have to purify the molecule. In this case, she purified the DNA. She set it up with an X-ray beam going through it, and basically, when the X-rays hit any of the atoms, the core of the atoms, they were deflected. And they make a specific pattern. And this X, it's known as photos 51, apparently, not to our untrained eyes, shows the repeating units of a helix. And Watson and Crick saw this at a seminar that she gave. Actually, it was Watson. And he went back to the lab to see Crick, and he said, "Hey, we got it. We figured it out." And they contacted Wilkins, who worked with Franklin. They had a little strange relationship. He thought they were equals. She thought he worked for him. That he worked for her. They shared lab space. They didn't love each other so much, but they worked together, is what I hear from biographies. And he actually shared her data with Watson and Crick when Watson and Crick came to visit him while she was out of town. So there is some hard feelings on Rosalind's side about how they got the information and how they published that first paper without their names, Rosalind's and Maurice's names on it. In any event, the structure of DNA looks like this. I'm going to zoom in here. Whoops, here. Lots of balls and sticks here. We'll see some more simplified things, but there's a couple of reasons to show this complicated picture. So the DNA is in this double helix. Here's one strand. It's going around the back, coming around here, and it's hydrogen bonded to the other strand. So DNA is said to be a double helix. When it does that and it spirals, has a slightly different spiral than the alpha helix we talked about with proteins. It leaves a major groove and a minor groove, and we're going to see that proteins take advantage of binding this when we have to regulate whether the DNA is going to be expressed and how it's wound up with proteins to be stored. A lot of interesting stuff. The distance between those two strands is very specific, and that was some information that the model building helped figure out. If you notice, the A's always bind with the T's and make 2 hydrogen bonds. The G's always bind with the C's and make 3 hydrogen bonds, just because of the way those bases get twisted. So an interesting thing is that, remember, the pyrimidines have just 1 ring. The purines have 2 rings. And because a purine always pairs with a pyrimidine, you maintain that distance in the helix. So it's a neat little feature because of the chemistry. I think we have another picture that's a little bit less intense. Let's zoom in on this one. Not this. Here we go. So let's get rid of some of the balls and sticks and kind of look at it a little bit more clear. So DNA is known as a double helix because 2 strands line up anti-parallel. Similar to the beta pleated sheets, how they went anti-parallel, one way, one the other way. 3' goes up, ends at the 5' end. 3' here, ends up at the 5' end. Hydrogen bonds holding them together. This shouldn't have 3 dots. This should have 2 dots, 2 hydrogen bonds hold the A and the T together, 3 hydrogen bonds hold the G and the C together. That's called the sugar phosphate backbone. That's what that blue ribbon is, and then the bases are the rungs of the ladder. RNA, that's what this picture is here, typically is found as a single strand, but it also has the ability to hydrogen bond and make different structures. So this is transfer RNA. This is involved in delivering the amino acids over to the ribosome to make a protein. We'll talk a lot about that later. There's ribosomal RNA that makes up the structure of the RNA. There's messenger RNA, which is the code to make the proteins, and we'll talk lots about that. And then there's other really fascinating RNAs. They're called small interfering RNAs. We'll talk about that when we talk about gene expression, and many more. Scientists think that RNA is the simpler one, and that's true. It's much less stable than DNA, so a lot of the new information, meaning the last 10 years, didn't come out until then, just because it was just so darn hard to work with the RNA. It degrades on the bench top when you're working it in the lab. And they think that RNA probably was the first molecule to evolve when we talk about the evolution of life later on. Zip through here. I like this picture. I left it in because I think it's fascinating. We have the crystal structure rendition of both a protein. So this is RNA polymerase 3. This is the enzyme that copies DNA into RNA. And you can see, let's find an alpha helix in there, beta pleated sheet in here, and a basic tertiary structure. It reads the DNA, helps it unwind. So here's our DNA molecule getting unwound. And then you can hydrogen bond or match the bases and synthesize a new RNA molecule. So this is a great picture showing all 3 together. So how are those 3 related? I'm going to jump. Well, we can start here. This is called the central dogma, and mostly because it was the first thing that people figured out. It still holds true, but now we know that there's exceptions. So DNA, whoops, didn't want to do that. Why is it doing that? DNA can be copied into itself using enzymes. DNA can get copied and made into RNA. DNA is a template. You make the RNA from it. And then RNA typically gets translated into protein. The RNA has a code that can get decoded by a ribosome, made into protein. We'll get into the details later. They call this special because we did not think that RNA could ever go backwards to become DNA, but retroviruses, initially other ones, but we know HIV is a retrovirus. It's an RNA virus that gets copied into DNA when it enters the cell. Those viruses taught us that this can happen. Special enzyme can do this, go in the reverse direction. We also know that it tends to happen in our cells with retrotransposons that we'll talk about in a later date, sort of as a holdover, and we're still figuring out what they're there for. And in some cases, RNA can get copied into RNA. But again, those are special cases. This is the flow of information that happens most of the time in the cell, so this is the central dogma idea. So DNA resides in the nucleus, double-stranded DNA. An enzyme comes in, and at the appropriate locations of a gene, copies that DNA into RNA. RNA then exits the nucleus as mRNA. A lot of complicated steps in between here we'll talk about in the next few months. That RNA gets found by 2 parts of a ribosome, the small subunit and the large subunit, and then that synthesizes the protein, the polypeptide, by putting the amino acids together. That sort of brings everything together. And I think that's it.